Hello friends, welcome to TechQuest channel. This is for the medical technologist by your tech. In this video, we will discuss about MacConkey Agar. MacConkey Agar was developed by Alfred Theodore MacConkey. He was a British microbiologist for Royal Commission on Sewage Disposal to report on what methods to disposing of sewage from factories or manufacturing process. He developed this medium for the growth of only gram-negative, non-fastidious organisms and to identify the bacteria with the lactose fermentation for his work. Bile salts and crystal violet makes the MacConkey agar as a selective medium. Lactose and neutral red makes the MacConkey agar as differential medium. Composition of MacConkey agar: peptone, bile salts, crystal violet, lactose, neutral red, sodium chloride, agar, and water. The pH is set to 7.1 plus or minus zero point 0.2. Let's see the importance of each component. Peptone. Peptones are short chains of amino acids linked by peptide bonds. A polypeptide is a long, continuous, unbranched peptide chains. Amino acids are basic requirements for the growth of bacteria. Peptone also provides nitrogenous nutrients for bacteria. Bile salts. Bile salts are used to suppress the growth of gram-positive and fastidious organisms. Bile salt also gets precipitated around the colonies when there is a growth. Crystal violet. Crystal violet or junction violet, also known as methyl violet, is a triaryl methane dye used as a histological staining methods and gram stain of classifying bacteria. Crystal violet has antibacterial, antifungal, antihelminthic, antitrypanosomal, and anti-tumor properties. Crystal violet was formerly an important tropical antiseptic. Crystal violet in small amounts in MacConkey agar used to inhibit the growth of gram-positive organisms. Bile salts and crystal violet in MacConkey agar makes it selective medium as the gram-positive bacteria are suppressed by these components. Next is lactose. Lactose is a disaccharide having galactose and glucose monosugars. This is a milk sugar, makes 2 to 8% of milk mass. In humans, lactase is the enzyme breaks down the lactose into galactose and glucose. In bacteria, lactose permease is the membrane protein symporter transport lactose to inside the cell. Lactose permease uses the protein gradient towards the cell to transport lactose in the same direction in the cell. The absence of lactose permease leads to the inability of lactose to enter the cell for further metabolic process. By mixed acid fermentation metabolic process, 6-carbon sugar is converted into a complex and variable mixtures of acids. It is an anaerobic fermentation reaction in bacteria. The mixture of end products produced in mixed acid fermentation are lactate, acetate, succinate, formate, ethanol, and the gases of hydrogen and carbon dioxide. First stage, the glucose is converted to pyruvate. During this stage, 2 ATPs and 2 NADP molecules are produced per 1 molecule of glucose. In second stage, pyruvate is converted to one or more products. In one product, the pyruvate is converted to lactic acid by lactate dehydrogenase enzyme. The lactose present in the MacConkey agar taken up by lactose fermenting bacteria by the lactose permease and fermented. The end product of lactic acid is excreted by the bacteria that changes the pH to acidic. The original pH of the media is 7.1 when prepared. Now the pH is lowered by the lactose fermentation. Neutral red. Neutral red is a vital stain and also a pH indicator. A vital stain can be used on living cells without killing them, like reticulocyte stain. Neutral red is also a pH indicator. It is a chemical detector for hydronium ions or hydrogen ions. The color of the solution changes depending on the pH. Neutral red is red in color at acidic pH and yellow in alkaline pH. The pH of the MacConkey agar is 7.1 when it is prepared. The bacteria capable of lactose fermentation produce the lactic acid at the end product that lowers the pH of the medium. In acidic pH, the colony changes its color to bright red to pink depending on the pH and the size of the colonies. Non-lactose fermenting bacteria cannot produce acidic product, so they remain at alkaline pH, looks yellow or colorless. Some of the bacteria produce very little amount of lactic acid, so the colonies appear yellow or colorless after 24 hours, and it changes to pink after 24 to 48 hours. Lactose sugar and neutral red together make the MacConkey agar as a differential medium to differentiate between lactose fermenting or non-lactose fermenting bacteria. Agar. Agar or agar-agar is a polysaccharide from the sea red algae. It has no nutritional value or interfere in growth of bacteria. It is used to solidify the medium. Agar melts at 100 degrees centigrade and solidify at 42 degrees centigrade. Sodium chloride. Sodium chloride is used to provide the electrolyte for the growth of the bacteria and maintains the osmotic equilibrium. The pH of MacConkey agar. The pH is the basic and important concept of MacConkey agar. The pH should be adjusted to 7.1 plus or minus 0.2 when the media is prepared. 7 is the neutral pH and it is prepared at neutral pH. The color of colonies depends on the acid product of the bacteria. Modified MacConkey agar. Many modifications are made to identify the selective organism's growth. The sugars or the bacterial inhibitory substances are changed according to diagnosis of the particular bacteria. These modifications are mainly used in water testing or food testing industries. Sorbitol MacConkey agar is used for particular E. coli strain growth. Double stem MacConkey broth is used in water purity testing for the growth of coliforms. Quality control on MacConkey agar. Escherichia coli, it should give pink color colonies with the growth when it is incubated at 33 to 37 degrees centigrade for 18 to 24 hours. Proteus mirabilis should give growth colorless colonies. Inhibition of swarming. Proteus vulgaris also gives growth colorless colonies but partial or total inhibition of swarming. Salmonella enterica cerovar typhimurium grows with colorless colonies. Enterococcus faecalis should not grow, it should inhibit the growth of this bacteria. Staph aureus also will not grow on MacConkey agar medium. Thank you.